Assalamualaikum and good afternoon, everyone. Welcome to our exciting webinar on the in silico approach to identify potential biomarkers and therapeutic targets in dementia patients. My name is Shamimi Shamsuddin, and I will be your moderator for today. This webinar is proudly organized by Unit Pendidikan Kesihatan, Pusat Perubatan USM Bertam, Universiti Sains Malaysia, and the Dementia Multidisciplinary Research Program of IPPT, DMR-IPPT. Before we dive further into the fascinating world of dementia research, let me introduce our esteemed speaker. We are honored to have with us Dr. Emmanuel Jairaj Moses, a senior lecturer from the Biomedical Sciences Department of IPPT. Dr. Emmanuel started his journey at USM in February 2016. Dr. Emmanuel holds a Bachelor in Biochemistry from Universiti Putra Malaysia, an MSc in Biotechnology from Universiti Sains Malaysia, and a PhD in Hematology from Universiti Sains Malaysia. For more details, please visit his profile on USM Expert, or you may reach out to him through his email. His extensive background and expertise make him the perfect guide for today's topic in our webinar. Thank you, Dr. Emmanuel, for joining us today. To kick things off, Dr. Emmanuel will explain what dementia is to us. And it is not just about forgetting where we put our keys. So, Dr. Emmanuel, what is dementia?
Dementia is, we understand dementia as a disease, but in actual fact, dementia is not a disease. Rather, it is a condition that is caused by several different type of diseases, as we will see in our talk later.
Okay. And it is common in our society to use this term called nyanyok. So, doctor, there is a common question. Are dementia and nyanyok the same thing? Let's clear up this confusion.
Okay, nyanyo is another word for nyanyo in English is senile, right? Senile. So it is different from dementia. Dementia is much more than just nyanyo. It has a broader meaning because it affects both the cognitive and also the psychological aspects of the brain.
Okay. Thank you, Dr. Emmanuel, for that introductory explanation. Now, we would like to dive further about dementia. What are the different types of dementia and which is the most common?
Okay, this question I will answer during my talk later. It will be part of my introductory slides.
Okay, so you mean it will be after we have gone through all the introductory questions, then you will start to share your slide, is it?
Yes, we can.
Okay, all right. Okay, does dementia only affect the elderly?
Okay, there is this common perception that dementia only affects the elderly. But as we will see in our talk later, dementia is, as I mentioned earlier, it is a condition. So it does not occur overnight. It is a process. So we will see later that dementia patients actually, they undergo different stages of dementia.
Okay, so are you saying that dementia not only affects the elderly, it can also happen
In
to
majority of the cases, the diseases that actually cause dementia, it affects the elderly. But there has been a growing research of evidence that shows that it is also affecting the younger generation, which is patients below the age of 65.
I
So
see.
this trend is actually quite worrying. Yes.
Great insights, Doctor. We really appreciate it. Now, closer to the title of the thought you're going to give later. What are the common biomarkers for dementia diagnosis?
Okay. These we will see in our talk also later. What are the common biomarkers that are available currently, and we will also have a brief look at what are the disadvantages of the current biomarkers that are available.
Okay, so by having the biomarkers, they do help to diagnose dementia.
Yes, the current biomarkers are actually quite accurate in diagnosing dementia, but they have certain disadvantages.
Okay, which you
So
will
we will,
cover in your talk. Okay.
yes, which I will cover in the talk.
Okay, this will include the weaknesses and also the disadvantages of the biomarkers currently available for dementia diagnosis. Okay, all right. So, Dr. Emmanuel, we are done with the warming up questions on our topic. So, without further ado, I would like to invite Dr. Emmanuel to deliver his talk on today's topic, in silico approach to identify potential biomarkers and therapeutic targets in dementia patients. And we will have a question and answer session after the talk.
Yeah, thank you, Dr. Shamimi, for that brief introductory session and also the brief Q&A session before the presentation. So let us move on to the presentation. Okay, a very good afternoon to everyone. Okay, so the title of my talk for today is, let me share my slide in a form of slideshow. Okay, everybody can see. Is it clear? Okay,
Yes,
the
Dr.
title of my talk for this afternoon is in silico approach to identify potential biomarkers and therapeutic targets in dementia patients. Okay, so this talk is a series of talk under USM Health Talk series, and it also covers the Dementia Multidisciplinary Research Program. Okay, as I mentioned earlier, dementia is not a disease by itself, but it is a condition which is caused by several types of diseases. Okay, so it is an umbrella term which is used to describe loss of memory and other thinking abilities, which is actually it has to be severe enough to disturb one's daily activity. Only then can we use this term dementia. Dementia most commonly occurs in patients with Alzheimer's disease. Okay, there are also several other types of dementia, which includes vascular dementia, Lewy body dementia, frontotemporal dementia, and there's also this mixed type of dementia where the cause is more than one, and also other type of neuro um, degenerative diseases such as Huntington's disease also causes dementia. What are the signs, some of the signs of dementia? So we can see here memory loss or subtle short-term memory changes. Then we have a difficulty concentrating and focusing, confusion with time or familiar places, misplacing things frequently, frequent mood changes, uh, failing sense of direction, and a difficulty carrying out normal routine activities. So as I mentioned earlier, I think in the introductory question, that there are various stages in the development of stages in dementia. So we have the first stage where there is no cognitive decline, and up to stage 3, there is a mild cognitive decline, and then stage 4 onwards, the disease, the condition progresses, and it ends in stage 7 where there is very severe cognitive decline. Okay. So what I would like to say is there is this early stage, mid stage, and also late stage of dementia, as we can see here. Okay, so this early stage, it is also sometimes known as MCI stage, or there is a mild cognitive inhibition. It is the early stage before dementia actually progresses. But we have to keep in mind that not all patients with MCI will progress to dementia, but they have the potential to progress to um, the more advanced stages of dementia. Okay. According to the DSM-5, the American Psychiatric classification, so this is the definition for dementia. So there has to be evidence of significant cognitive decline, as mentioned earlier. So this cognitive decline has to interfere with independence in everyday activities, meaning that even to do simple tasks, it is a major hurdle for dementia patients. Okay. And it is, although there are overlapping symptoms, it is separate and distinct from delirium. Okay. And also other major depressive disorders as like schizophrenia. So currently, how is dementia diagnosed? So there is no single test that can actually confirm dementia. So a dementia diagnosis is a multi-step process. So the diagnosis involved is full blood count tests, renal panels, and these all the biochemical tests. And then there is also ECG, neuroimaging by using CT and MRI scans, and also PET scan where indicated. And using this diagnostic tool, the diagnosis of dementia is actually quite accurate. Okay? But however, we must understand that by the time all these investigations are actually performed or done, usually the patient will already be at least at the moderate or advanced stage of dementia itself. So it is actually quite difficult to actually catch this condition at early stage, quite an early stage. So that is where we have these biomarkers. Biomarkers are early screening tools that are used to catch a condition or disease early on when it is at the initial stage. So that is the use of biomarkers. The currently available biomarkers for dementia, it is accurate, that is for sure. So, but nevertheless, a major disadvantage of having the current dementia biomarkers are these biomarkers are number one, as you can see, you need advanced equipment because you need to look at the specific brain area for atrophy. So meaning you need to do some MRI and CT scans and also PET scans. And then looking at tau deposits and amyloid beta deposits in the brain. This also it is quite an invasive procedure, and we can see that they also measure the levels of the amyloid beta and tau lipids, microRNAs, and these are done by drawing this cerebrospinal fluid. Okay. Cerebrospinal fluid, it is quite difficult to obtain. Number one, you get it in very low quantities, and number three, as I mentioned earlier, it is also invasive. So this is the major disadvantage of the current biomarkers that are established and available for dementia patients. Okay. Due to these invasive procedures, usually these tests are not really carried out at the early stage. Okay, so many a times dementia is missed out during the early stages of progression. Okay, it is not only about early detection. We also can see that the current biomarkers that are available for dementia, it only can detect the disease, like for amyloid beta and tau, by using the cerebrospinal fluid. It also can determine the disease risk by using this apolipoprotein E, and staging can be done by tau using looking for tau deposits using PET scan. So PET scan is not so readily available, and it is also quite expensive to be done. But we can see that for dementia, you only can determine the disease risk, you only can detect the disease, and you only can do staging. And even then, you need advanced equipment for it. Okay. Currently, there's no biomarker that is available to monitor the progression and recurrence, or I would say monitor the progression of dementia, because dementia is a progressive disease. And then there is also no biomarker to monitor the treatment adherence or to predict the treatment response. Okay, and then these are the gaps that are available where we talk about dementia biomarker itself. Okay, there are many aspects that are not fully covered as today. Okay, the era of omics has brought us many great technologies that enable the detection of many types of biomarkers at the genomic level. So we have under RNA, the transcriptomics, we have next generation sequencing for RNA-seq, and then we can do single cell sequencing and also microRNA, microarray, and also small RNA sequencing for the non-coding RNA. And then we have whole genome sequencing, whole exome sequencing, single cell sequencing for the DNA, genomics and epigenomics. And then we also have proteins, proteomics. We have MALDI-TOF MS, 2D gel, and electrophoresis, and many types of other technologies to detect the biomarkers at the proteome level using proteomics. And in the realm of metabolomics, we have mass spectrophotometry and NMR spectroscopy to detect the various metabolites that could serve as biomarkers. But for today's talk, I'm only going to look at the RNA transcriptomics, and I'm only going to focus on mRNA. So we are going to talk about just the RNA-seq data. Okay, so in this study, what was done was we tried to use this tool known as GEO2R. So GEO2R is a tool that is available on this GEO database. GEO database is Gene Expression Omnibus. I will let you all look at the interface later on. So this is just a brief summary. So what was done was using this search database, using the keywords dementia, RNA-seq, and human, 297 RNA-seq data was found. And then from there, all single cell RNA-seq data, mouse models, PDX model data were omitted. Why is it, you may ask me? Because we want to screen the entire transcriptome for potential biomarkers. So for this case, single cell RNA-seq would not be that suitable because single cell RNA-seq it is very compartmentalized, so it will give you information on specific areas of the brain instead of giving you a whole view of the what is going on. And furthermore, for differential gene expression analysis later on, RNA-seq is literature has pointed out that RNA-seq is much preferable to single cell RNA seq data itself. So based on this, this data were omitted, and finally we were left with 9 RNA-seq data. So this 9 RNA-seq data was analyzed by this GeoToR application, which is available on the Geo database itself. I will show you later. And to identify the top upregulated and downregulated genes based on the volcano plot. And based on the volcano plot, only 4 RNA-seq data sets were identified. I will show you all why is that again in the results. And these data from RNA-seq data sets were filtered and sorted on a Galaxy platform. This is another bioinformatics platform to identify top, up, and down-regulated genes. And these top upregulated and down-regulated genes were displayed on the SR plot. Okay, next. Okay, this is how the geo or gene expression omnibus interface looks like. If you can see, I would like to bring your attention to other tools. Other tools, we can see the 1, 2, 3, the 4th line, we can see analyze a study with GeoToR. So these were the tools that were used in the initial part of the study. And this is the volcano plot that was generated. I would like to attract your attention to see that when we look at volcano plots in bioinformatics analysis, volcano plot should look like this, where there is in the center, there's very dense areas of and as moving out, just like a volcano, we have scattered amounts of genes. So the red on the left side, it shows up regulation, and the blue it shows a down regulation. So this 4 is a nice pattern of a volcano plot that we will obtain from RNA-seq data. Okay, so these 4 were out of the 9 data sets. 4 data sets gave these very nice volcano plots. The other 5 did not, and I will show you why. So there's this representative 2 that I picked from the 5 that were not nice. We can see here why is the reason. We don't see any dense centers where there is blue or red genes, or no, but then rather we just have a few scattered. So when we obtain this kind of volcano plot, it is not ideal for us to do further gene expression analysis or differential gene expression analysis. Okay. So next, filtering was done using this tool known as Galaxy. So we sorted and filtered the data. So there is a whole series of pipeline that you have to follow for you to select your genes, and this tool is very important for you to actually explore and to further. I'm not going to show this all today because it's going to be a very long talk. Otherwise, I just want to just summarize everything. Okay, so what I did was, once I filtered and sorted, and I got the when I put it on the SR plot, I managed to get the top regulated and non-regulated genes from my RNA-seq data sets. So the top regulated genes, there were 24 that I managed. So this is the first 8, the second 8, and the third 8 that I managed to get for the upregulated genes. And then for the downregulated genes, I got 6 of it from the bioinformatic analysis. And then, so what I did was I listed down the top 6 upregulated and unregulated genes based on the p-value ranking that was obtained. So the lower the p-value, the more accurate the data. So these are the top 6 upregulated and downregulated genes that were obtained. And so what I did was I just want to know a rough picture or get a rough clue on the gene functions of these genes roughly. So I use this tool known as a G profiler, where it displays the gene ontology. The gene ontology, we have MF, which is the molecular function, BP, biological process, and then we have KGG, which is the molecular pathway, together with the wiki pathway and reactome. Those are all molecular pathway. And then, so what we can see is in the upregulated genes, mostly they are involved in this metabolic processes, inflammatory processes, and also DNA damage repair pathways. So we can see that that these upregulated genes actually cause roughly it causes DNA damage. It causes dysregulation of metabolic processes, and it also causes induction of inflammation, which actually contributes to the neurodegeneration in general. Okay, this is for the upregulated genes. Now for the downregulated genes, I'd like to attract your attention here, especially to when we look at the molecular function, we can see that this galactose proton symporter activity, it is reduced. So it has something to do with the disruption of the blood-brain barrier here. Because as we know in neurodegenerative disorders, the blood-brain barrier is actually compromised. So we can see that this is the one of the main biological processes that are affected, as well as signaling pathway. We can see defective mismatch repairs. And then what I would like to attract your attention to is let us look at the HPA, Human Phenotype Association, or the HP, the human phenotype. Here we can see abnormality of mental function, memory impairment, and it maps back to the various parts of the brains. So meaning this in silico analysis that is done, it is very accurate in the sense that it maps back to the phenotype. The genotype matches back to the phenotype that is being displayed. Okay, so this is one way of how you know that you actually fishing out these potential biomarkers. Okay, you have to do the analysis of the gene annotation and the metabolic pathways or other regulatory pathways to actually just try to get a general picture of the gene function. Okay, so this is just at the beginning of the story. Identifying those markers. One thing interesting is if you look through literature, these genes that are listed here, they have not been reported anywhere else. If you look through literature, these genes have not been reported anywhere. Okay, so this is just the beginning of the story. What needs to be done further is we need to validate the expression levels and the gene functionality of these genes that have been selected in ex vivo, in vitro, and in vivo preclinical neuronal models. So the work is far from over. So we cannot say that whether these are potential biomarkers or therapeutic targets. So we have to validate and we have to study the function before any conclusion can be drawn. Okay. And next, what we need to do is we need to try to analyze this expression level in easily accessible bodily fluids such as saliva, urine, and other types of bodily fluids to make, because one of the challenges of these biomarkers is the invasive method of gathering the samples. So what we need, research is currently carried out to actually make this process less invasive. So what is currently being done is the expression levels are being measured in bodily fluids that are readily available. Okay. And so we need, because this RNA-seq data, most of it was obtained from these tissues, post-mortem brain tissues. So we need to try to measure the bodily fluids and see whether the genes can be detected, the expression levels of these genes can be detected, because we know that in most of the neurodegenerative disorders, the blood-brain barrier is actually compromised. So these genes might be circulating in the circulation as well. Okay. So as before I end the talk, I would like to attract your attention to the future direction that people are looking for in this biomarker discovery. Number one, they are looking for less invasive biological fluids that I've spoken about. They are looking for minimally invasive methods, okay, low cost, ease of sample collection, and improved sensitivity and accuracy in that is where these enhanced biosensors in the form of aptamers and antibodies, graphene, and carbon nanotubes also come into play. Okay, so this is the future direction of how biomarker discovery is going to go. Okay, instead of more invasive procedures that are currently being done, less invasive methods are being studied with low costs and also improve accuracy and sensitivity, and to which end biosensors could actually serve as an important tool as well, um, and together with the omics technology that is currently available. Okay, that's all. Thank you.
All right. Thank you so much, Dr. Emmanuel, for the insightful presentation. Now we will open the floor for our question and answer session. You have two options to participate. You can either unmute your microphone if you like to ask your questions directly, or you can use the WebEx chat box. You can type your question and I will read out your question for Dr. Emmanuel to answer. Let's get started. Who would like to go first for the question and answer session? Okay, Dr. Emmanuel, maybe I could start first for this session. Okay, for the future direction in this field of research, you did mention just now the application of enhanced biosensors. So which field of engineering can actually collaborate together for this particular innovation to take place for dementia research?
Okay, so it depends on which type of biosensors we are talking about, whether it's a chemical-based biosensor or whether it's an electrical-based biosensor. It depends what we are trying to use to detect. So I would say that materials engineers would be useful to actually characterize the material itself. And then we have these electrical electronics engineers to help in the circuit or wiring, and also the chemical engineers to look at the impedance and many types of other parameters that are needed to be resolved in the biosensors before we can proceed. So it is actually a multidisciplinary or transdisciplinary approach where we talk about biosensors.
All right. Thank you for the explanation. So I would like to invite our participants to voice out any questions to our speaker today. All right, Dr. Emmanuel, seems like we don't have any questions. Oh yes, there's one from Mr. or Dr. Adam Azlan. Is it okay if I read it out?
Yeah, sure.
Okay. Hi, Dr. Emmanuel. Since detection depends on earlier diagnosis, is there a particular process that a patient needs to undergo before being labeled as dementia or diagnosed as dementia positive?
Okay, thank you, Dr. Adam, for this question. So before we can actually diagnose a patient with dementia, only a psychiatrist actually can diagnose a patient with dementia. So the biomarkers that I was talking about would only serve as an early screening tool for you to actually perform the further diagnostic tests. And this only can be done by a psychologist. And it has to be a multidisciplinary approach where you need to use a questionnaires. You also need to do the more advanced techniques of looking at the areas of the brain using MRI and so on. So, but biomarkers would just serve as a early screening tool, right? So when you have something that you can detect earlier on, then you can also, the intervention can also begin earlier on.
All
And
right.
as we know, dementia is not, it is a progressive disease, so we cannot cure the condition. I mean, we cannot cure the condition, but we only can halt or delay the process.
Yes. Okay. All right. Thank you, Dr. Emmanuel and Dr. Adam Azlan for the question. We are still waiting for questions from our participants. Okay, there's one more. As patient progresses, do the levels of these markers change?
Okay, for the current biomarkers that are currently available, like amyloid beta and tau, yes, the levels do change. But as for the newer biomarkers, research is currently being done because there are, if we look through literature, there are like several candidate microRNAs that have been identified to serve as potential biomarkers. But this is actually still under research and it has not really been established as of yet.
Okay, all right. Thank you, Dr. Emmanuel and Dr. Adam. All right, any more questions from our participants? As I have mentioned at the earlier part of our webinar, this webinar is organized by PPUSMB, USM, and also the Dementia Multidisciplinary Research Program of IPPT. So we highly encourage researchers who are interested to collaborate with us to reach out and we can collaborate together in this study on dementia. Okay. All right, Dr. Emmanuel, looks like that's a wrap for our webinar today. Do you have any concluding remarks before we end?
Yeah, I would like to also echo what Dr. Shamimi says. You can always reach out to this Dementia Multidisciplinary Research Program for future collaborations. And if anybody is interested in pursuing this in research in this field, can always contact Dr. Roayu and Dr. Shamimi or any one of the team members. And
Alright.
thank you very much for sparing your time, your precious time to join this talk. And thank you, Dr. Shamimi, and also Dr. Roayu, and also Puan Akida for arranging all the logistics and everything else. Thank you very much.
Thank you so much. As we wrap up, I would also like to thank Dr. Emmanuel for his invaluable insights and to all the participants for your participation today. Soft reminder to everyone, please remember to fill up the Google form so that you may obtain CPD marks for attending today's webinar. Let's end our webinar with a pantun before we say goodbye. Kalau ada jarum yang patah, jangan disimpan di dalam peti. Kalau ada silap dan salah, jangan disimpan di dalam hati. Thank you everyone. Stay curious and keep learning on dementia. Thank you. Bye.